Hello viewers, today we are going to learn about what is gene cloning. So gene cloning एक ऐसा प्रोसेस है जिसके अंदर हम अपना जीन ऑफ इंटरेस्ट जो है वो इंसर्ट करते हैं इनटू द वेक्टर सो डैट वो जो मेरा जीन ऑफ इंटरेस्ट है वो उसकी मल्टीपल कॉपीज जो है वो बना सके. लेट अस सी हियर. Gene cloning is the process of producing multiple identical copies of a specific gene by inserting into a suitable vector and allow it to replicate inside the host cell. Now let us try to understand. So ये एक वेक्टर है. वेक्टर का मतलब है एक ऐसा जीन जिसके अंदर मैं अपना जीन ऑफ इंटरेस्ट को इंसर्ट कर सकूं और वो होस्ट सेल के अंदर जाकर मल्टीप्लाई हो सके. डैट इज़ नोन एज द वेक्टर. वेक्टर इसको हम ऐसे व्हीकल जैसे भी समझ सकते हैं कि जैसे अगर मुझे कहीं पर भी कौन सी भी डेस्टिनेशन पर जाना होगा, to reach that destination, I need the vehicle. In the similar way, to insert the gene of interest, it needs the vehicle, and that vehicle is known as the vector. Clear? Now, this is the vector, and I am having the gene of interest, and this is my gene of interest, and this gene of interest has to be inserted inside the vector. So here the gene of interest is inserted inside the vector. Now this vector is known as the rDNA. We will learn more about rDNA in our next slides. Clear? Now this rDNA is able to move inside the host so that this particular copy would be multiply. So this is basically the gene cloning. Now, what are the steps which are involved in the gene cloning? So first step that is involved is to isolate the plasmid from the vector. Clear? And second was the DNA fragment containing the gene to be cloned is inserted into a circular DNA which is known as the vector to produce rDNA molecule. What is rDNA molecule? It is the recombinant. अपना जीन ऑफ इंटरेस्ट को इंसर्ट किया तो ये क्या बन जाएगा? rDNA. मीन्स एक DNA के अंदर मैंने एक और DNA को इंसर्ट किया, दोबारा से इंसर्ट किया, दैट इज नोन एज द recombinant because it is the combination of both the DNA, gene of interest and the host DNA. So it is the combination of two DNA and two DNA is inserted. So that is known as the recombinant. The vector transport the gene into the host cell. Clear? When the host cell multiplies, it also going to multiply the our gene of interest. Second was when the host cell divides, the copies of the rDNA also divides. Clear? Now let us try to understand from this diagram. In this diagram, what is being represented here is that it is having the bacteria from which I am going to isolate this plasmid. Plasmid I am going to isolate from the bacteria, and this is my gene of interest, and this region is particular my gene of interest to which I need the multiple copies. that is the gene of interest. Now moving ahead, now the plasmid is isolated from the bacteria and it is having my gene of interest in this long fragment. Now, very important step is both, that is rDNA and my gene of interest, should be cut with the same type of the nuclease enzyme. So, what is nuclease enzyme? Nuclease enzyme is act as the molecular seizure. सो सीजर का काम क्या होता है? किसी भी चीज़ को कट करना. सिमिलरली वो जो हमारे जीन्स को कट करती है, डैट सीजर इज नोन एज द मोलिकुलर सीजर. सो ये जो मोलिकुलर सीजर है, ये क्या करेगी? मेरे इन डीएनए फ्रेगमेंट्स को क्या करेगी? कट करेगी. और ये कट कैसे करेगी? अगर ये क्या होंगे? सेम होंगे. सेम molecular restriction enzyme से we are going to cut our both plasmid and our gene of interest. Why so? Why we need to cut with the same type? So that it is going to produce either the blunt ends or the sticky ends. Sticky end means that overhangs. Clear? And blunt end means that both are having same type. क्लियर? तो हमने क्या करना है? या तो हमको सेम रिस्ट्रिक्शन एंजाइम से वी आर गोइंग टू कट इट. लेट अस से दैट आई एम गोइंग टू कट at this side and this is my gene of interest. So here I am using endonuclease restriction enzyme. So endonuclease restriction enzyme comes and makes the cut over here. Here the endonuclease restriction enzyme comes and makes the cut over here. So when the cut is made, it is going to produce the sticky ends. Here also sticky ends, here also sticky ends. So sticky end is being produced. Happened is that you can see that here the sticky end and here the sticky end is produced. Now there is the work of the ligase enzyme. Now this ligase enzyme come and is going to bind both the fragments of the plasmid and my gene of interest. So when there is the binding of the plasmid along with the gene of interest, that is known as the rDNA. rDNA means recombinant DNA. So recombinant का मतलब क्या होता है? दोबारा से. कॉम्बिनेंट का मतलब क्या होता है? कॉम्बिनेशन. बिकॉज मेरे पास ये पहले से ही डीएनए क्या था? प्रेजेंट था. अब मैंने इसके अंदर अपना एक जीन ऑफ इंटरेस्ट जो कि एक फ्रेगमेंट थी किसकी? डीएनए की, उसको मैंने इंसर्ट कर दिया. दिस इज दी न्यू डीएनए. और ये जो मेरा था ओल्ड डीएनए जो था वो मेरा पहले से ही प्रेजेंट था. ठीक है. अब यहाँ पर कॉम्बिनेशन हुआ है किसका? दो डीएनए का, एक न्यू वन और एक ओल्ड वन. और जब भी कॉम्बिनेशन होता है न्यू और ओल्ड का, उसको हम क्या बोलते हैं? rDNA. That is known as the rDNA. So what is going to happen next is that now this rDNA is ready to get insert into the host cell DNA. Now this is the chromosomal DNA and my rDNA is inserted inside the host cell. So once it is going to inserted inside the host cell, the cell is going to divide, and alongside the division my gene of interest is also getting multiply. So when we are going to get these multiple copies, this process is known as the cloning. As we know that DNA is basically divided by the binary fission. So this is my DNA and having my gene of interest inserted there. Now this bacteria is going to divide by the binary fission. So binary fission means when the cell is going to divide into the two equal parts. So when it is going to divide into the two equal parts, each part having my gene of interest, and this is also going to divide. So here also my gene of interest produce, here also gene of interest produce, and when it is going to divide here also. So multiple copies of my gene of interest is produced only by this process, and that process is known as the cloning. I hope you are better understood. So this is all about in today's lecture. In upcoming lectures, we are going to study what are the properties of the plasmids, because not every plasmid is able to transform our gene of interest into the host cell. So there are some properties which is only present in the plasmid. So this we are going to study in our upcoming lectures. Thank you.